Hi everyone, and welcome to our video on the topic of respiration. So this video we will go through from the beginning to the end of the topic of respiration to keep unit 2. Okay, so just a little outline as to what this video will have in it. So we'll be going through the 4 main steps in respiration, which would be first glycolysis, then the link reaction, which is not essentially a big part of it, but is a very important part. Then we'll be going through the Krebs cycle, and finally we have the electron transport chain. But that's not all we'll be doing. We'll be also doing anaerobic respiration, which you would have heard about already, and investigating the rate of respiration with an experiment. Alright, so that will be our agenda for today's video. Alright, so let's jump into glycolysis one time. Okay, so I'm going straight to this diagram here. Alright, so glycolysis occurs in the cytoplasm of the cell. That's very important, right? It occurs in the cytoplasm of the cell. Okay, so there are a few steps in glycolysis. You can see from this diagram, we have A, B, C, D, E, and F as well, right? So these are very important. So in A, you can see that the glucose enters the cell. So that's the cell membrane there, and glucose enters the cell. So that's the first step of glycolysis, right? The glucose has to enter the cell via diffusion, right? And then once it's in the cell now, you can see that glucose goes to glucose 6-phosphate and then fructose 1,6-bisphosphate by similar reactions using ATP. So you can see ATP here is going to ADP, ATP is going to ADP. So 2 ATP are actually used up. And remember, the process of respiration is to make ATP, so we better be making some profit after spending some ATP there, right? So these 2 steps are called phosphorylation. And remember, the change of ATP to ADP is called phosphorylation, right? So phosphorylation occurs twice, right, in B and C. Okay, now once there's fructose 1,6-bisphosphate, the next step is lysis. So lysis, the word lysis means to split, right? So fructose 1,6-bisphosphate is actually split into 2 molecules called triose phosphate, right? So they are the same molecules, triose phosphate. So now each triose phosphate molecule will undergo the same reaction. So essentially all the reactions are now doubled. Okay, right. So in step E, we have oxidation. Right, so the triose phosphate molecule is oxidized to this intermediate bisphosphate. It's not essentially essential to learn what that molecule is. It's just an intermediate molecule before we get to our final molecule, right? But in order for it to be oxidized, NAD needs to be reduced. So the opposite would happen to NAD. And remember, everything happens twice, right? So you end up with 2 reduced NAD molecules there. All right. And then in F now, we have substrate linked phosphorylation. All right, so again, we have ADP changing to ATP. All right, so as you can see, the substrate linked phosphorylation occurs twice for each side. All right, so you have ATP being manufactured, another ATP being manufactured. So that's 2 on that side and then 2 more here. Okay, so therefore we manufactured 4 ATP in glycolysis while we used 2 ATP up here for a net profit of 2 ATP molecules. And that's not all we get in in glycolysis, we also get these reduced NADs, which will come in handy later on. Alright, and you'll see why very soon. Alright, now the end product of glycolysis is just very important molecule here, pyruvate. So we also get 2 pyruvate molecules, and that goes straight into the link reaction. Okay, so before the Krebs cycle can begin, pyruvate needs to be converted essentially, right? So in the link reaction, you can see it here, pyruvate reacts with something called coenzyme A plus an NAD molecule to achieve this very important molecule here, acetyl coenzyme A. Plus you get more reduced NAD and you get some carbon dioxide. Okay, not very important here. The important things to note are the acetyl coenzyme A and the reduced NAD. So we got reduced NAD in glycolysis, and now we are getting more reduced NAD in the link reaction. Okay, now acetyl coenzyme E is very important in helping the reaction of a certain molecule called oxaloacetate, which we will see in the Krebs cycle. Alright, and that essentially starts the Krebs cycle. Without that, it could never occur. Okay, so this little table here down here says for each glucose molecule after the link reaction, we get 2 carbon dioxide. You don't need to know about that, but we get 2 reduced NAD and 2 acetyl coenzyme A molecules. So therefore, remember, everything that occurs after glycolysis occurs twice, right, for each pyruvate molecule, right? So therefore, when we go to the Krebs cycle, let me go to it here, there are actually 2 Krebs cycles occurring per glucose molecule, because remember, the glucose molecule is split into the 2 triose phosphate molecules, and you have 2 pyruvates, right? So all these steps occur twice. Alright, but we'll have one big summary as to how many ATP molecules we are making per glucose molecule, right? So we add up all basically. Right, so that was the link reaction. As I said, not too big there. It's just to make the acetyl coenzyme A, right? So let's go to the Krebs cycle. Now the Krebs cycle, unlike glycolysis, occurs in the matrix of the mitochondrion, right? Glycolysis occurred in the cytoplasm of the cell. Okay, right, so now we have acetyl coenzyme A here, and we have oxaloacetate. Oxaloacetate is a 4-carbon compound, acetyl coenzyme A is a 2-carbon compound, so they are both going to react to make this 6-carbon compound called citrate. Okay, so the important things to learn here in the Krebs cycle are the yellow boxes, right? We need to learn off these molecules here. You don't need to learn the purple enzymes. Okay, that's irrelevant basically. But the blue stuff and the ATP stuff you also need to know, as well as these green stuff down there as well. We'll get to that. Okay, right, so that was the first step, oxaloacetate combining with acetyl coenzyme A to make citrate. The second step is very simple. Citrate converts to isocitrate. It's basically the same thing, just an isomer of the other. Okay, now isocitrate in the third step undergoes a dehydrogenation and decarboxylation reaction. That might sound complicated, but that's actually the same thing that happened to pyruvate in the link reaction. Let me show you. So pyruvate was actually decarboxylated, meaning it lost a carbon molecule, right? And it lost it via carbon dioxide. And it was also dehydrogenated because an NAD became reduced. Now, reduced NAD is also called NADH, as in it accepted a hydrogen molecule. So the pyruvate was also dehydrogenated, meaning it lost a hydrogen. It was also decarboxylated, meaning it lost a carbon. Okay, so nothing too big there, right? So the isocitrate, we say it's dehydrogenated and decarboxylated. Right, and that forms alpha ketoglutarate, right? This one down here. Now, whenever something is dehydrogenated and decarboxylated, it loses carbon dioxide. But again, we are not concerned about that. You just breathe that out after you respire, right, as a waste product. But what we do like is the reduced NAD being formed, or NADH, right? So that's another one. So you could see in the process of respiration, a lot of these NADHs pop up, right? Right. So let's continue now. Alpha ketoglutarate down here actually undergoes more decarboxylation. All right. So this undergoes the same thing as in 3, right? So this is step 4. Right, so it's decarboxylated to succinyl coenzyme A, and every time we decarboxylate, we produce a NADH. So we like that. All right, you will see why that's very important afterwards. Okay. Now succinyl coenzyme A in the 5th step forms succinate. However, it's not a decarboxylation reaction. It's actually substrate linked phosphorylation, which we saw in glycolysis, right? So in substrate linked phosphorylation, we make an ATP molecule. So that's very important. So you see, that's the first ATP molecule we made in the Krebs cycle. Okay, right. So now we are at succinate, kind of in the middle of the Krebs cycle there, right? Now succinate goes to fumarate, right? And this is simply oxidized. However, there's no NAD over here. Instead, to go to fumarate, we use FAD. It's not very important to know exactly the difference between NAD and FAD, but just know that it's reduced in the same way as NADH. It's just FADH2. That's just how the molecule is, right? But it's the same process. It's reduced. Okay. So we add fumarate now. Fumarate is simply converted into malate by adding water. Not very important there. And finally, in the 8th and final step, to get back to oxaloacetate, malate is dehydrogenated, right, to form oxaloacetate. So again, now we make another NADH molecule and we go back to oxaloacetate, and there we will start over the Krebs cycle again, right? So this is the cycle, 8 simple steps. You just simply need to learn how to draw it, practice to draw it, practice to learn off your molecules, and you'll get there, right? So just a quick summary as to what was made in this Krebs cycle. We have 1 NADH, 2 NADHs, we have a FADH, and we have 3 NADH molecules. So we have 3 NADH, we have 1 FADH2, and we also have the ATP molecule, 1 ATP molecule. Now this is per pyruvate, right? Every pyruvate molecule produces these 3 things. However, per glucose, remember, glucose makes 2 pyruvate, right? So per glucose molecule, we will have 6 NADHs, 2 FADH2, and 2 ATP molecules, right? So all these are produced in the Krebs cycle for 1 glucose molecule. So now let me show you why NADH and FADH2 are important. Now we are at the final step in respiration, called the electron transport chain. Right? It looks a little bit complicated, but it's not that bad. I will show you what I mean now. So in the molecules NADH and FADH2, right, they consist of high energy electrons because they were just made, right? So the electrons in these molecules are high energy. So in the electron transport chain, every orange molecule here are called carriers, right? Electron carriers. So we start off with NADH reductase, which is the highest energy electron carrier. So all the high energy electrons from NADH and FADH are going to pass through NADH reductase and step down the chain like if they're going down a ladder. And every time the electron goes down a rung or moves to another molecule, they lose energy. right? So the electrons lose energy in the form of ATP, right? So the electrons have a lot of energy to give. So as they go down the ladder, they keep giving off ATP, keep giving off ATP. And we like that. And then finally, so it goes down more and more carriers, right? So cytochrome is a popular carrier until it eventually binds to oxygen of all things. And then it's being converted to water and it will be excreted in the body, right? So before it's excreted, it makes a lot of ATP, right? And this is actually the whole purpose of breathing in oxygen in the body. For just one reaction here at the end of the electron transport chain. If you noticed, for the whole of the topic of respiration, we didn't see an oxygen molecule yet. You might be wondering, so well, what's the purpose of oxygen in respiration? This is the process here. This is the only purpose here of oxygen, just to get rid of the electrons at the end. Okay, so that's what the electron transport chain is about. Now, the different molecules, NADH and FADH2, they give off different energies, right? So, essentially, for one NADH molecule, one NADH molecule gives off 3 ATP. These electrons are very high energy, okay? One FADH2 molecule, they give off 2 ATP molecules, okay? So just keep that in mind as to when they have to go through the electron transport chain. So knowing this now, we can actually calculate, well, how much ATP is actually made in respiration per glucose molecule, okay? So this is called the energy yield for aerobic respiration, okay? So we have glycolysis, the link reaction, the Krebs cycle, and the electron transport chain. Okay, so let us do ATP first, the actual ATP, right? So in glycolysis, we made 4 ATP molecules there, right? Now we not take into consideration the fact that we used well, we could actually do it as in the net production of ATP, right? So remember, we used 2 ATP in the beginning of glycolysis, but we made 4. So the net product of ATP in glycolysis is 2 molecules, right? In the link reaction, we didn't have any ATP being made. We only made the NADH molecule, right? So in the link reaction, we made no ATP, right? In the Krebs cycle, each Krebs cycle, we make 1 ATP molecule. And remember, per glucose molecule, we actually have 2 Krebs cycles. So in the Krebs cycle, we make 2 ATP molecules there, right? And in the electron transport chain, well, it kind of depends on the amount of NADH and FADH molecules there. So we'll leave that out for now. Let's take a look at the NADH molecules being produced, and then we'll calculate the electron transport chain, right? So in glycolysis, we actually have a good bit, right? So let's see how much we have in glycolysis. If we read the passage here, we can see that there are 2 NADHs being formed in glycolysis, right? So that's actually per triose phosphate molecule, right? Because if we go up back to the glycolysis, you can see that there's one reduced NADH here, another reduced NADH here. So per glucose molecule, we actually have 2 NADHs being formed, right? So let's go down here and we could put 2 NADH. Now I have to put the FADH2 here, although there's nothing being in glycolysis. It's just for the Krebs cycle, right? Now the link reaction also has 2 NADH molecules being made, right? Let's go back up and check that. So here we have the link reaction. You can see we have a reduced NADH here, and that's per pyruvate molecule. So for the glucose molecule, we have 2 pyruvate molecules, so there's actually 2 reduced NAD being formed there. Alright, so let's go back down. So we have 2 NADH molecules there. Now the Krebs cycle is responsible for making quite a lot of NADH molecules. So let's count them. We actually did the calculation already here. Right, so we have a lot of things being formed here, and that is per glucose molecule, right? So per glucose molecule, we make 6 NADH and 2 FADH molecules. All right. So in the Krebs cycle, we make 6 NADH molecules and 2 FADH molecules. Right. And now we can actually use these two to calculate how many ATP the electron transport chain will make. So per NADH molecule, we get 3 ATP molecules from. So we have 2 from glycolysis, 2 from the link reaction, 6 from the Krebs cycle. That is 10 NADH molecules. So 10 by 3. I'll actually put it here. 10 by 3. That's 30 ATP there, right? And then for the FADH2, we get 2 ATP per molecule. So we have 2 of them. So that's 2 by 2, which is 4. Right? So in the electron transport chain, we get a total of 34 ATP molecules. So to total it up now, the total amount of ATP in respiration per glucose molecule is 38 ATP. So we get 2 from glycolysis, 2 from the Krebs cycle, and 34 from the electron transport chain. So that's how we get 38 ATP per glucose molecule in aerobic respiration. Okay, and that is the bulk of respiration there. So let's just do a quick rundown of anaerobic respiration. So in anaerobic respiration, organisms can make ATP without the presence of oxygen. So which means that there's no electron transport chain being involved, there's no Krebs cycle, it's really just glycolysis. Okay, so without oxygen, we cannot regenerate these, the NAD and FAD. So you have to do something else to try to get them back, right? So the reactions of glycolysis, you know, makes 2 ATP molecules. And organisms can survive on 2 ATP molecules for some time, right? Depending on what the organism is, okay? So in order to try to regenerate the NAD and FAD, which you know is very important, the pyruvate at the end of glycolysis is actually converted to something else depending on the molecule, right? So the pyruvate is said to be decarboxylated to ethanol, right? And this is in yeast. It's also called alcoholic fermentation because of the presence of ethanol, right? And the enzyme that does this is pyruvate decarboxylase. So you can see here, the glucose goes to pyruvate. Just this is glycolysis here. We don't need to go back through the whole glycolysis, right? And then pyruvate now, in the yeast cells, undergoes alcoholic fermentation. It makes ethanol, and then that converts to ethanol plus carbon dioxide, and then you get energy. Right, so the carbon dioxide in alcoholic fermentation is what makes bread rise as well, right? So that is an application of alcoholic fermentation in yeast. That's how bread is made, right? That's how bread rises. Now let's take a look at anaerobic respiration in animals. So it's very similar, right? Except what happens to pyruvate at the end. Okay, so in certain situations where you want ATP to be made quite rapidly, right, you would depend on anaerobic respiration. And you know about this since Form 5, right? So glycolysis can happen very quickly without having to go through the whole electron transport chain, which takes quite some time, right? So even though it would supply 2 ATP molecules. It will take place quickly, and since NAD will be regenerated, it can keep going, right? The glycolysis can keep going, because remember, glycolysis, you need NAD, right? So in order for this to occur, the pyruvate at the end of glycolysis here will undergo lactic acid fermentation to make lactic acid. It has to do this in order to regenerate the NAD molecules, right? That's what will keep the glycolysis going. However, lactic acid alters the pH in the blood, right? And remember, you need your correct pH for your enzymes to function. And it is because the pH changes, the enzymes will not function as they usually do, and this is why you will feel tired after exercising rapidly for some time, right? Now, we can't have this lactic acid buildup, so the body will eventually get rid of it, and to do this, it occurs in the liver, right? And it actually has to reoxygenate the lactic acid. So your body has to give oxygen to the lactic acid to get rid of it. And this is actually called the oxygen debt. This is why after exercising for so long, you will have to breathe deeply, and you have to take some deep breaths in order to catch back yourself. That is called the oxygen debt. Alright, so the final thing we'll be doing is actually looking at an experiment to measure the rate of respiration. So in this experiment here, we have this scale that is made up of a ruler, right? And we have a little liquid droplet in the ruler. So what will happen essentially is that air will push through the tube and will push the droplet to one side, right? And the more air that is pushing the droplet, the faster it will move, and the more we could calculate the rate, right? What you have in this set of the apparatus here, right? So you have your small organisms that will be undergoing respiration. You have the soda lime, right? So the soda lime has a very important function, right? So the soda lime will absorb the carbon dioxide that is being produced by the organisms, right? So you can't have the carbon dioxide pushing the droplet as well, because then it would not it would lead to errors in the experiment, right? So the soda lime there absorbs all of the carbon dioxide. Now the gauze is to simply separate the organisms from the corrosive effects of the soda lime. If you put the organisms in soda lime, they will just die, right? So the gauze there is important to separate the organisms from the soda lime. Okay, so in this apparatus we have the air here, as I told you, right? So air is all over here, right? Now remember, respiration, aerobic respiration, uses oxygen. So all this and all this over here is air that is available for these organisms to use. So therefore, if they are undergoing aerobic respiration, they will be using oxygen, and they will actually suck the this droplet this way, right? And remember, they will also be producing carbon dioxide, which would have pushed the droplet back, right? Because it would be like you use air, but you're still breathing out air, right? That is the important function of the soda lime. So the soda lime will absorb the carbon dioxide. So therefore, the oxygen will be used up, and the organisms will not be putting out air to push back the liquid, right? So remember, if they're sucking the liquid like a straw, if you're sucking the air out of the tubing like a straw, the liquid is going to follow it because of the negative pressure, and you will simply have to look at where the liquid was first, where on the ruler it is, and then after a certain amount of time you measure where it is now. So let's say after a few minutes it reaches here, so you measure from here to here, and you will see the distance that the liquid moved. You will have the time that you took for the liquid to move, and therefore you could calculate the rate, right? The rate is like speed, right? So therefore you use that way to calculate the rate of the reaction. It's a very simple experiment, a very easy one as well. So you just need to learn how to draw it and the functions of the important parts of the apparatus, right? Meaning the liquid, the ruler being the scale, the soda lime to absorb the carbon dioxide, and the fact that the organisms will be breathing in oxygen, which is only available here and through the tubing, right? So as they use the oxygen, it's going to pull the liquid, and there you will get your measurements. Okay, so I hope you guys enjoyed this video. I tried to make it as short as possible because respiration is a long topic, so you could go through it how many times you want and make sure you grasp topics. Okay, bye everybody.